Hello, everybody. My name is Miyah Brooks. I'm in the Nigerian Immunity Lab under Dr. Nikki, Traylor, Knowles, and I'm going to be doing my research on pharmaceutical approaches to coral bleaching immune modulation during heat stress. So when we have a fever and our body starts to overheat, that's our immune system activating, letting us know that something is wrong, right? When we commonly experience these symptoms like a headache or a fever, we would take something like an ibuprofen or some type of anti-inflammatory to reduce the inflammation and bring our bodies back to balance. Just like us, corals have their own way of signaling when something is out of balance, letting them know that they need to fix it. Corals form a mutualistic relationship with their microscopic algae that provides their energy, but environmental stressors such as heat, light, and pollution can disrupt this partnership. And when that happens, corals expel their algae, leading to known as bleaching and energy loss. But we also know that heat stress upregulates immune-related genes, including pro-inflammatory pathways in many coral species. But when we look at where research is actually focused, shown in the figure, we see that it's mostly centered around stress response and symbiont physiology, while immunity makes up only a small fraction of this research. But what if coral bleaching is not just stress, but immune response? More specifically, what if inflammation plays a critical role? This leads to my question, can anti-inflammatory drugs reduce bleaching under heat stress? To address this, I have two aims. First, I want to determine whether immune modulation changes bleaching under heat stress, and second, I want to understand how different types of anti-inflammatory drugs affect coral immune responses. To test this, I use two types of anti-inflammatory drugs. One, ketorolac, which is a non-steroidal anti-inflammatory drug that reduces inflammation by inhibiting prostaglandins. And two, I use dexamethasone, which is a steroid that strongly suppresses immune activity by targeting key inflammatory pathways. These two drugs will allow me to compare different levels of mechanisms of the immune suppression. I then expose corals to elevated temperatures under controlled conditions, and at the same time, I treated them with these drugs to modulate their immune response. To measure this response, I tracked the corals' symbiotic algae, both that remained in their tissue and the ones that got expelled out during bleaching. I assessed this through using imaging, cell counts, flow cytometry, and protein assays. This allows me to directly determine whether altering the immune response changes the bleaching process. Ultimately, this work will show whether immune modulation affects coral bleaching under heat stress. More broadly, it helps shift how we think about bleaching from something that just simply happens to corals, but something that may be actively regulating through their immune system. Because just like a fever signals and lets us know that our immune system is working hard, coral bleaching may be that internal immune response letting them know that something is wrong. So if we can understand that signal and key better, we can predict and help protect these coral reefs in a warming world. Thank you.